Hello everyone, welcome to BioQuest Biochemistry classes. Today I am back with a new video tutorial on Hexose Monophosphate Shunt. This is a common topic in MBBS exam as a LAQ, SAQ, and this is also a hub for MCQ in various entrance examinations. Now the objectives. This video tutorial will help the viewers to understand the salient features of Hexose Monophosphate Shunt, steps of the pathway, key regulatory steps of this pathway, importance of the glucose 6-phosphate dehydrogenase enzyme, and the questions relevant to this topic that has appeared in various entrance and MBBS examinations. So without much more delay, let us start the video tutorial. The salient features of Hexose Monophosphate Shunt. This pathway is also known as the Pentose Phosphate Pathway or the phosphogluconate pathway. It occurs in the cytosol of both prokaryotes and eukaryotes. It consists of two phases, oxidative phase and the non-oxidative phase. No ATP is consumed or produced in this pathway. This is a remarkable feature where the sugar has been metabolized yet no ATP is produced or required for the metabolizing of the carbohydrates. Important products produced in this pathway are NADPH and ribose, which is a pentose sugar, and it has an aldose functional group that is required for the synthesis of the nucleotides. The C1 of glucose 6-phosphate is liberated as carbon dioxide and 2 NADPH are produced for each molecule of glucose entering the Hexose Monophosphate Shunt. Now, there is a common question in the Viva why it is called a shunt. This is because the oxidative pentose pathway is so named. It involves some reactions of the glycolytic pathway and therefore it has been viewed as a shunt of glycolysis. That's why it is termed as HMP shunt. The rate and direction of the reversible reactions of the pentose phosphate pathway are determined by the supply of and demand for intermediates of the cycle. Moreover, this pathway also produces ribose phosphate that produces a mechanism for the metabolic use of 5-carbon sugars obtained from the diet or the degradation of structural carbohydrates in the body. Now, let us look at the phases of the first phase of the HMP shunt is the irreversible oxidative phase. Here, the glucose, the hexose sugar will be acted upon by the enzyme hexokinase and it will involve the utilization of one molecule of ATP and it will be converted to ADP and glucose will be converted to glucose 6-phosphate. This requires hexokinase. Now, some of you may get confused that initial slides I have said that it does not require ATP. This is the first pathway of glycolysis 2. So, this has directly no relation to the HMP shunt. Now, this glucose 6-phosphate will be converted to 6-phosphogluconolactone and here the NADP will be converted to NADPH plus H plus. The enzyme for this reaction is glucose 6-phosphate dehydrogenase. This glucose 6-phosphate dehydrogenase, it is also the key regulatory enzyme of the hexose monophosphate shunt, and both glucose 6-phosphate dehydrogenase as well as hexokinase require the bivalent cation magnesium. Now the 6-phosphogluconolactone will be acted upon by the enzyme lactonase and it will be converted to 6-phosphogluconate, and this conversion requires hydrolysis by a molecule of water. This enzyme also requires the bivalent cation magnesium. 6-phosphogluconate dehydrogenase will act upon the 6-phosphogluconate and it will be converted to 5-carbon keto sugar known as ribulose 5-phosphate, and here again NADP will be reduced to NADPH and one molecule of carbon dioxide will be liberated. So have a look at the reaction. Glucose, which was an hexose aldose sugar, will be converted to 5-carbon keto sugar, that is ribulose 5-phosphate, and one molecule of carbon dioxide is liberated in this step. Now this ribulose 5-phosphate will get isomerized to aldose 5-carbon sugar ribose 5-phosphate, and this 5-carbon sugar will form a major source for the formation of or biosynthesis of nucleotides. This whole pathway, the reactions involved in this step are in the oxidative phase of the HMP shunt. So the end product of the oxidative phase of the pentose phosphate pathway is the ribose 5-phosphate. Now let us look at the non-oxidative phase. Now 2 molecule of glucose, if it undergoes the reactions of the oxidative phase, 2 molecules of ribulose 5-phosphate will be produced. Now this ribulose 5-phosphate will again get converted to its epimer xylulose 5-phosphate and it will again get isomerized to ribose 5-phosphate. So from 2 molecules of 5-carbon ribulose 5-phosphate, I am getting 1 molecule of its epimer xylulose 5-phosphate and another molecule of ribose 5-phosphate. Now these two again they will act together and this reaction between the two 5-carbon sugar will lead to the formation of 3-carbon aldose sugar, that is glyceraldehyde 3-phosphate, and 7-carbon keto sugar, that is known as pseudoheptulose 7-phosphate. Now if we have a look at this reaction, the enzyme required is transketolase. The transketolase require thiamine pyrophosphate as the coenzyme, and the estimation of erythrocyte or RBC transketolase activity is a surrogate marker for the assessment of vitamin B1 status in an individual. Moreover, thiamine pyrophosphate is key regulatory coenzyme for the pyruvate dehydrogenase complex E1, for the alpha ketoglutarate dehydrogenase of the TCA cycle, and the branch chain alpha keto acid dehydrogenase required for the metabolism of branch chain amino acid. So the transketolase that requires thiamine pyrophosphate, this thiamine pyrophosphate is also serving the purpose of coenzyme in various other reactions. Now again there will be shifting of the aldose ketose group between the glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. What we will get? We will get 6-carbon fructose 6-phosphate and 4-carbon aldose sugar erythrose 4-phosphate, and the enzyme which is catalyzing this reaction is known as transaldolase. This transaldolase, it transfers 3 carbon units and it converts the glyceraldehyde to 6-carbon fructose phosphate. Now again the ribulose 5-phosphate may get converted to xylulose 5-phosphate, and this xylulose 5-phosphate may react with the erythrose 4-phosphate to form fructose 6-phosphate as well as glyceraldehyde 3-phosphate. So all of this fructose 6-phosphate, glyceraldehyde 3-phosphate, they are the important intermediates of the glycolytic pathway. The link of the video tutorial on glycolysis have been given in the description box or in the I button. You can find the link of this video tutorial. That's why we can see that many metabolites in the non-oxidative phase are indirectly linked to the glycolytic pathway. That's why HMP shunt is also the other name for the pentose phosphate. Now the key regulatory step of HMP shunt. The key regulatory enzyme of the HMP shunt or pentose phosphate pathway is the glucose 6-phosphate dehydrogenase, and this glucose 6-phosphate dehydrogenase, this enzyme is regulated by the NADP plus NADPH ratio and the oxidized or reduced glutathione. When NADPH is forming faster, then it is being used for biosynthesis of glutathione reduction, because we will see that this NADPH is the coenzyme for the enzyme glutathione reductase. So if NADPH is less utilized, then the reserve of NADPH will increase and the ratio of NADP plus NADPH will decrease, because NADPH is in the denominator and it will inhibit the first enzyme in the pentose phosphate pathway. As a result, more glucose 6-phosphate that will be available for glycolysis. It won't enter the HMP shunt, it will be channelized to the glycolytic pathway. Moreover, there is another important question, that what are the other sources of cytosolic NADPH? Remember the mnemonic HIM, H for Hexose Monophosphate Shunt, I for isocitrate dehydrogenase that is present in the cytosol, and M for the cytosolic malic enzyme. These 3 enzymes, or rather 2 enzymes and 1 important metabolic pathway, are the important sources of the cytosolic NADPH. Importance of NADPH. It is important for the reductive biosynthesis. Reductive biosynthesis requires this NADPH. NADPH, the high energy molecule, it behaves much in the same way like the NADH, but the electrons of the NADPH are destined for use in the reductive biosynthesis of steroids, cholesterol, fatty acid, rather than transferred to oxygen as in the case with NADH, which enters the electron transport chain. Thus, in the metabolic transformations of the pentose phosphate pathway, part of the energy of glucose 6-phosphate that is conserved as the NADPH molecule can be used in reaction requiring a high electron potential electron donor. So NADPH is important for reductive biosynthesis of cholesterol, steroid hormone, fatty acids. It provides reducing equivalent required by the cytochrome P450 monooxygenase system. Now, monooxygenase, as we all know, they are also known as mixed function oxidase. Why they are so named? What is the reason? Because this mixed function oxidase incorporate one atom from molecular oxygen into a substrate, creating a hydroxyl group, while the other atom being reduced to water. Moreover, in this cytochrome P450, the P stands for pigment and 450 stands for the wavelength 450 nanometer, at which it shows the absorbance maxima. There are two important cytochrome P450. One is in the mitochondrial system, another is in the microsomal. The microsomal system of cytochrome P450 is associated with the phase 1 reaction of the metabolism of xenobiotics, and the mitochondrial cytochrome P450 monooxygenase system participate in the hydroxylation of steroids like 11 alpha hydroxylase or 17 alpha hydroxylase, which is important for the synthesis of the steroid in the adrenal cortex. They are the part of the mitochondrial system. That's why if we look at the places where the oxidative pathway takes place at higher rate, this of HMP shunt, this portion of the pathway is particularly important in liver, lactating mammary glands, adipose tissue, because they are important reservoirs for fatty acid synthesis. In the adrenal cortex, where NADPH is required for biosynthesis of the steroid hormones, and in the erythrocyte to keep the glutathione in reduced form. Phagocytosis of white blood cells. The white blood cells that phagocytose the bacteria, they kill the bacteria by two mechanisms. One is via the oxidant dependent mechanism, another one is the oxidant independent mechanism. NADPH is essential for the oxidant dependent system. Now, if we imagine this is a phagolysosome, the oxygen that will be acted upon by NADPH oxidase and it will be converted to superoxide anion. The NADPH oxidase is an important enzyme, and if this enzyme is deficient by birth, it leads to chronic granulomatous disease. Now, this superoxide will be acted upon by the superoxide dismutase and it will be converted to hydrogen peroxide. This hydrogen peroxide will react with the chloride anion and it will form the HOCl. This catalysis is taken care of by the enzyme myeloperoxidase. Now, this HOCl will act on the bacteria and will cause lysis of the bacterial cell. And NADPH is also important for the synthesis of the nitric oxide. The L-arginine is converted via the enzyme, a flavoprotein enzyme, nitric oxide synthase to L-citrulline, and nitric oxide is formed. This nitric oxide, it is an important neurotransmitter, as well as this nitric oxide is also important for the vasodilation in our vascular system. Previously it was termed as EDRF, endothelium derived relaxing factor. Moreover, NADPH is also essential for maintaining the glucose 6-phosphate dehydrogenase enzyme. This enzyme is the key regulatory enzyme of the pentose phosphate pathway. Moreover, this enzyme is the most common enzyme deficiency associated with hemolytic anemia. The most common enzyme whose deficiency leads to hemolytic anemia is glucose 6-phosphate dehydrogenase enzyme, followed by the pyruvate kinase. Now, this enzyme is the deficiency is inherited in X-linked recessive pattern. That means the females are the carriers and the male get affected. Now, it is seen that the highest prevalence of this enzyme is seen in Middle East, tropical Africa, Asia and parts of the Mediterranean. Now, why it is so important? Because this enzyme deficiency normally does not present with hemolytic anemia. But if there is some stress or some drug that causes some oxidative stress, the patient's presents with hemolytic anemia. Now, this is a classical presentation of the peripheral blood smear of a glucose 6-phosphate dehydrogenase enzyme associated hemolytic anemia. We can see the bite cells are there in the peripheral smear, typical as if someone has taken a bite of the RBCs. Moreover, His bodies will also be seen in the peripheral blood smear in the erythrocyte of the patients having G6PD deficiency. Now, what are the drugs which are very notorious to precipitate hemolytic anemia in patients having G6PD deficiency? This is remembered by the mnemonic of SNP. S for the sulfur containing drug like sulfonamide, sulf-methoxazole and dapsone. N for nalidixic acid, nitrofurantoin and naphthalene balls. It is a classical case that a baby was playing very well and his mother was shuffling the winter garments. The baby has put one naphthalene ball in his nose, and after 1 hour the mother comes to the OPD. The baby is having cola colored urine. So this is a classical presentation of glucose 6-phosphate dehydrogenase deficiency. Isoniazid, that is an antitubercular drug, and primaquine, that is used to prevent the relapse of malaria. This enzyme, if it is deficient, these drugs may precipitate hemolytic anemia. That's why before prescribing primaquine, it is mandatory to screen for glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase, this enzyme is expressed in units like unit per gram of hemoglobin or unit per cubic millimeter of RBC. For the enzyme estimation, we have to send the blood in EDTA vial, and simultaneous hemoglobin and RBC estimation is necessary for interpretation of this enzyme. Now, why there is this enzyme deficiency precipitates with hemolytic anemia if the drug is given. Now, the oxygen that will be acted upon by the NADPH oxidase, it will be converted to hydrogen peroxide. It will be acted upon by the superoxide dismutase and it will form H2O2. This H2O2 will get converted to water and it is acted upon by the selenocysteine containing enzyme glutathione peroxidase, and here the reduced glutathione is converted to oxidized glutathione. The most potent antioxidant molecule in our body, the glutathione. Now we have to again regenerate the reduced glutathione, and for regeneration of the reduced glutathione, we need glutathione reductase enzyme. Now this glutathione reductase enzyme is it requires the coenzyme NADPH that is produced from the pentose phosphate pathway, and the key regulatory enzyme of the pentose phosphate pathway is glucose 6-phosphate dehydrogenase. Now if glucose 6-phosphate dehydrogenase is absent, There will be no NADPH, no reduced glutathione will be regenerated, and the RBC will be exposed to the free radical injury by the hydroxyl free radical, and we will see that the hemolysis occur. That's why glucose 6-phosphate dehydrogenase enzyme screening is mandatory before prescribing antimalarial drugs like primaquine, because if the enzyme is deficient, such drugs will cause the free radical injury to the cell membrane, and they will precipitate the episodes of hemolysis. These are the important questions. Enumerate the oxidative and non-oxidative phase of HMP shunt. Why pentose phosphate pathway is so named? Why G6PD deficiency leads to hemolytic anemia? An important product of pentose phosphate pathway, that is the ribose or the ribulose 5-phosphate and the NADPH along with their. Thank you for watching. Stay tuned for next videos. Thank you.